This lecture is on the urinary system. The urinary system consists of several organs. It consists of the kidney, the uterus, the bladder, as well as the urethra. So the two kidneys, the two ureters, one bladder, as well as the one urethra. Okay. Do not confuse ureter with urethra. Okay. We have two ureters, hence the 2E. Whereas there is only one E in urethra, so we only have one urethra. Okay. All of these together is called the urinary system. And the kidney specifically functions to help filter things out of our blood. And that includes nitrogenous waste, toxins. You can read these other ones yourself. Okay, and in doing so, it also helps ensure that our body is able to maintain a chemical composition, so the right amount of electrolytes, for example. Okay, and having the right amount of electrolytes in our body also helps ensure that there's adequate fluid. There's a proper amount of fluid retained inside our body. Finally, the things that are excreted through your urinary system, these are metabolic waste, and all of these together are known as nitrogenous waste. So these nitrogenous waste, respectively, are urea, uric acid, and creatinine. You're probably going to hear more about these terms when you get into physiology. Okay, so kidney does most of these jobs, and then the job of ureters, bladder, and urethra really is just transporting urine out of the body. But the bladder serves as a temporary storage of your urine before you go use the restroom, and then it's peed out. First, let's talk about the gross anatomy of our kidneys. Okay, I'm using old slides here to give you additional illustration. Your kidneys are the two bean-shaped organs that lie in the retroperitoneum. So behind the peritoneum, you're the posterior part of your abdominal cavity. And in terms of their level relative to the vertebral column, they are near the end of your rib cage, the inferior portion of your rib cage. Specifically, this region is called the costovertebral angle. I want to note it here. You can write it down in your notes. Here, the CVA stands for, I just want to mention that you're not responsible for knowing everything in these slides. Focus on what's in here, but I'm just making a note on here right now because there's more white space over here. Okay, so the CVA here stands for costovertebral angle. Okay. So this is the location, approximate location of your kidneys. Okay, it's more posterior than anterior, as you can see in here. Okay. And costovertebral angle, CVA tenderness, is a term used to describe oftentimes as a sign of kidney infection. So when someone gets kidney infection, they have back pain in this region. You can tap on it and you can see tenderness there. Okay. Like many of our other organs, your kidneys are wrapped around by layers of tissue. The fibrous capsule surrounds immediately around the kidneys. And around that, there are the perirenal fat capsule. Okay. And then there are other supportive tissue layers, the renal fascia, which I don't want you to worry about right now. It's not something we're going to go over in this class, actually. Okay. But I do want you to understand the structure of the kidney. Okay. This outer region is known as the cortex. So you see the region that's outlined by my pointer. This region is known as a cortex. On a test, you should write renal cortex. If you only write cortex, well, cortex, as you're going to see, is a term we use to describe parts of different organs. There's adrenal cortex, right? There's a cortex in your brain. So there's different structures where we use cortex with. But specifically, this is renal cortex. Renal is referring to kidneys, right? But make sure you understand that urinary and renal are not the same. Urinary, okay, is referring to the whole urinary system. Renal is specifically referring to the kidneys. This is the renal cortex. This inner region is called the renal medulla. Renal medulla. The renal medulla consists of renal pyramids. So the pyramids are these triangular shaped or cone shaped, right? Because you have to remember that this is only a section of the kidney. Looking at this in 3D, this will be cone shaped. So this will be an individual renal pyramid. Now the renal sinus is a cavity within the kidney, which is occupied by the renal pelvis, the renal calyx, and blood vessels, as well as nerves and fat. So this dipped in region on the surface of the kidney. The renal pelvis specifically is this wider region of your ureter, so this dilated portion of your ureter, where the renal calyces merge, okay, and before the urine drains into the ureter. Now, as for a renal hilum, you can think of renal hilum as the entrance to the renal sinus. So sinus is this dipped in region, right? And the hilum, remember when we talked about hilum of your lung, right? It's this bundle of things. So this is the entrance way into this dipped in region called the renal sinus. This is something that you do have to unfortunately memorize, which is the order or the sequence of blood flow through these structures involving the kidney. So it begins in the aorta, abdominal aorta, which then branches to the right and left renal arteries. Each of the renal artery, the right renal artery and also the left renal artery, they further branch into segmental arteries. Segmental artery then branch into the interlobar arteries. Interlobar arteries are between the different renal lobes. Okay, so I want to clarify that this is a single renal lobe. This is a single renal lobe. So a single renal lobe includes a single pyramid with its associated cortical region. Okay, so renal lobe, renal lobe, renal lobe. Okay. So between the lobes, that's where those interlobar arteries are located. So after interlobar arteries, these interlobar arteries then branch to form the arcuate artery. The arcuate arteries, as its name indicates, it form an arc, okay, right between the junction between the renal cortex and the renal medulla. So this is where those arcuate arteries would be. Each of the arcuate arteries then branch into cortical radiate arteries. So if you look at the name, you understand why it's called cortical radiate. This is the renal cortex, correct? They radiate into the renal cortex, hence the name cortical radiate arteries. Okay, and to look at this microscopically, okay, we see that each of the cortical radiate arteries then branch into afferent arterioles. So if you look here really closely, you can see that's those little spines. Okay, those are the afferent arterioles. Afferent arteriole, also known as afferent glomerular arteriole, then lead into a structure known as glomerulus. Glomerulus is really just a specialized type of capillary. Okay. And the things that are not filtered into this thing called a nephron will continue on into the efferent arteriole. The blood continues into the efferent arteriole. Okay. Efferent arteriole then leads into peritubular capillary beds. Okay. The peritubular capillary beds, we have a picture here. So here's a simplified version of it. But here's another schematic for peritubular capillaries. These are capillaries that wrap around the structures, renal tubules. Okay. And after that, these vessels can drain their blood into either vasa recta, okay, or it can drain directly into these cortical radiate veins. So this is something you do have to look into closely, because I understand it can be kind of confusing when you first go through it. Okay. And then from there on, the cortical radiate veins lead into the arcuate veins. Okay, cortical radiate veins lead into arcuate veins. Arcuate veins lead into these interlobar veins. Okay. There is no segmental veins. So then from the interlobar vein, okay, that just leads into the renal vein. The renal vein then leads into the inferior vena cava. Okay, so this diagram shows you the structures that the blood flows through in that right order, in that correct order. So passive blood flow through the renal blood vessels. Okay. We're going to talk about some of these details later, and you'll see why later. It's because I want to follow the structures in the order in which the blood as well as the urine and filtrate flows in. So we're going to first look at the microscopic anatomy of your kidneys. If we zoom into your kidneys, we'll find the basic structural and functional unit of your kidney called a nephron. You have about 1 million nephrons in each of your kidneys. So with the 2 kidneys present, that's about 2 million nephrons. And these are the workhorses for filtering your blood. Okay. And the way in which they carry out this process of filtering your blood and modifying your blood content is through 3 processes: glomerular filtration, tubular reabsorption, tubular secretion. Okay. And here's what each one means. At the level of the glomerulus, the structure is called the glomerulus. Okay. This glomerulus determines what in the blood can be allowed into the nephron. Okay. So if it passes into this glomerular capsule, it will continue through the renal tubule, and eventually it may end up in the urine. But on the way there, the content of this filtrate is still being modified extensively. Okay. It may undergo tubular reabsorption, meaning the contents gets reabsorbed. Okay. And some contents can, even by the time it gets into the peritubular capillary, it still has a chance of being excreted or secreted out of your blood into the renal tubule. Okay. That's why as long as it's before the end of these renal tubules, we still call this content a filtrate. We don't call it urine. Okay. By the time it's exiting out of the collecting duct, okay, that's when we start calling this urine. Okay. Looking at the specific anatomy of each nephron, individual nephron. So here's a anatomy of the nephron. There are several parts to this nephron. Okay. You can break nephron down into renal corpuscle, okay, as well as renal tubules. Okay. So the renal corpuscle, I'm not sure where it says. Okay, so this is supposed to be. Okay. Renal tubule and renal corpuscle. Okay. Now the renal corpuscle is this structure right here. Renal tubules is these. Okay. So the nephron is made up of renal corpuscle and renal tubules. The renal corpuscle can be further broken down into the glomerulus, which is the capillary, the tuft of capillary, specialized capillaries in there. And the glomerular capsule is this external capsule that wraps around the capillary. Okay, so those are the two parts of renal corpuscle. The renal tubule can then be further broken down into the proximal convoluted tubule, okay, also known as PCT. On the test, you should spell it out. The PCT will be oftentimes the abbreviation that you can see. PCT follows into the nephron loop, also known as the loop of Henle, okay, right here, LOH, which then lead into the distal convoluted tubule, or DCT. Convoluted is referring to its the shape of these structures. Okay. DCT of different nephrons will merge into the same collecting duct. Okay. And then for the loop of Henle or nephron loop, there's a descending part and then there's an ascending part. So notice where these tubules are located. Okay, so if this portion is the renal cortex and below this junction, this is the renal medulla, you should see that looking at the kidney, okay, this portion is where we see the renal corpuscle, as well as the proximal convoluted tubule and distal convoluted tubule to be located. And the loop of Henle as well, so the loop of Henle or the nephron loop would expect them to dip down into the renal medulla. Taking a closer look at the renal corpuscle. So this is a cross section of the renal corpuscle. We see the afferent arteriole, also known as afferent glomerular arteriole, and efferent glomerular arteriole, or efferent arteriole. You can just write efferent arteriole, afferent arteriole on the exam. It's fine. I will accept that. Compare their size. You notice that afferent arterioles are thicker, or they're wider than the efferent glomerular arterioles. And our little preview into physiology, which we'll study next semester, is that the width difference, okay, creates a positive pressure, hydrostatic pressure, that pushes, promotes water to be filtered, okay, and substances to be filtered through this filtration membrane. Okay, and you see how this structure, once our afferent arteriole enters into this renal corpuscle, okay, it becomes more convoluted. It forms a structure called the glomerulus. The glomerulus is just a tuft of the arterial capillary, shown in red, but wrapping around the glomerulus are these special cells called podocytes. These podocytes are really cute cells with spiny feet, and their feet forms slits. Okay, and we'll see a picture of the podocyte slits in just a second. Okay, but I also want to show you during the first unit when we talked about the simple squamous epithelium of these nephrons. Okay, this is what we were saying, right? So this is the glomerular capsule, also known as Bowman's capsule. Bowman's capsule. Okay. There are two layers to this Bowman's capsule. This is the parietal layer, which is made up of simple squamous epithelium. Okay. And then the visceral layer, is just the podocytes. So the podocytes are associated intimately with the fenestrated capillary of the glomerulus. And you can see here in terms of histology, the epithelium transitions into simple cuboidal epithelium once we enter into the renal tubule. Now, if we're to take a cross-section of this filtration membrane, this is what we are going to expect to see. This is inside the glomerulus, and this is inside that capsular space. Okay, so for a substance to enter the capsular space, it has to leave first the capillary, so it has to first be able to exit out of the simple squamous epithelium that makes up the glomerular epithelium. Okay. And then it has to cross the basement membrane. The basement membrane just has connective tissues in there. And then it has to then be able to pass through these filtration slits formed by the feet of those podocytes. The pod is referring to the feet. So these cells are characterized by their spiny feet. So these are foot processes of the podocytes. Okay. As you can see, conditions that damages either the podocyte feet or damages the capillary wall here will allow more substances to be filtered into the capsular space than normal. Right? Okay, if this is really damaged, you can actually get blood cells, which normally doesn't leave the space to exit into the capsular space. And that's how you can end up with blood in the urine. Okay, which is a side note, not something you have to worry too much about right now. Okay, so this structure formed by the capillary wall, okay, so the simple squamous epithelium, the capillary endothelium, the basement membrane, as well as the foot processes of the podocytes, these three layers make up the filtration membrane. I do want you to understand that there are only certain molecules that are allowed to pass through the filtration membrane. Generally, only the small molecules like water, ions including sodium, potassium, chloride, okay, glucose, small proteins, okay, as well as a nitrogenous waste like urea are allowed to pass through into the capsular space. Things that are kept inside the blood are the blood cells, like white blood cells, red blood cells, okay, platelets. Those are too large under normal circumstances to pass through the filtration slit. That's why they stay inside your blood, okay. Now that we just talked about the first process in urine formation, which is filtration, so it's called glomerular filtration because the filtration is occurring at the level of glomerulus. Okay. The two steps that follow after are tubular reabsorption and tubular secretion. Okay, and these two processes are both occurring throughout the renal tubule. Okay. So it's not that renal absorption necessarily happens before secretion. Both of these are happening at the same time. So those tubular reabsorption, tubular secretion are occurring at the level of the renal tubules. The proximal convoluted tubule, okay, PCT, located inside the renal cortex, just like DCT, they're both located in the renal cortex. Okay. There are specializations that allow it to be able to effectively reabsorb as well as secrete. So this should really read reabsorb, absorb. Okay. I've crossed out the parts that you don't have to worry about. Even though the histology, the type of epithelium somewhat changes down the loop of Henle, but all I want you to know is that for the most part, your renal tubules are made out of simple cuboidal epithelium. So for the most part, it's simple cuboidal epithelium. That's all you have to understand in terms of histology of renal tubules. And then the renal corpuscle has the Bowman's capsule. The Bowman's capsule, its parietal layer is made up of simple squamous epithelium. Okay. So sorry, I got sidetracked. But moving from the PCT into the loop of Henle, or LLH, the LLH consists of a descending limb and an ascending limb. So you should be able to identify which one is ascending, which one is descending. Okay, descending comes first, following the path of urine, not the urine, sorry, following the path of filtrate. Remember that we don't collect urine until the end of collecting duct, right? Because the composition of this filtrate is still undergoing much modification throughout the tubules. It's still far from what eventually is going to become the urine. So here the filtrate enters into the descending, is moving down. So this is a descending limb and an ascending limb. Descending limb is the one that's closer to the PCT. And then the DCT is the one that leads into the, the descending limb is the one that leads into the DCT. Okay. So throughout this process, there are things that are being reabsorbed and then secreted. Okay. The loop of Henle in particular is very important in concentrating of urine, making urine more concentrate. Much of the ions that gets filtered into the Bowman's capsule, like something like 90% of the sodium, I can't remember the exact number, but much of the, I think it is actually closer to 99% of the sodium that gets filtered into the Bowman's capsule actually gets reabsorbed by the end of PCT. Okay. You have to understand that there's a lot of ions that gets filtered into the Bowman's capsule, and then large majority of it is reabsorbed back into the body, back into the bloodstream first, right? Along with water. So that helps your body retain water. And the details of that, I don't want you to worry about until you get to physiology. Okay. So throughout this process, there's reabsorption and then there's secretion happening through the renal tubule. The collecting ducts right here, it does not carry much of reabsorption or secretion, but its main function is in responding to this hormone secreted by your brain called ADH. ADH stands for antidiuretic hormone. And there's another term for it. I should have included in here. It's called vasopressin. Vasopressin. Okay. Vasal is referring to the vessels. Pressin is referring to blood pressure. Okay. So this is a hormone that's capable of binding to the collecting duct. And what happens is when those hormones get to the kidneys and bind to the collecting duct, it causes collecting duct to open up water channels and then allow lots and lots of water to be reabsorbed back in your body. And when there is more water inside your blood, guess what happens to your blood pressure? Your blood pressure then goes up. So that's what the collecting duct is important for. It responds to antidiuretic hormone. Notice that it does not secrete antidiuretic hormone. This is a hormone that's actually secreted in your brain. Okay, but it flows through the blood vessels and then gets to the kidneys, to the collecting duct, and binds to the collecting duct, and it induces a response here, okay, causing increased water reabsorption. Okay. And the effect of that is once you pull that water out of collecting duct, it causes your urine to become more concentrated. Okay. And it contributes to conservation of your body fluid. So at any given time, there is some ADH secretion by your brain, but when you're dehydrated, your ADH level is going to go higher, and because that's your body trying to reabsorb as much water as possible. Okay. Sorry, I may have gone into a little more detail that you're going to learn about in physiology, but focus on what is here on the slide. Okay. Now, there are two types of nephrons. There are the cortical nephrons, and that's the large majority of nephrons. And there is a minority of nephrons, okay, that's called juxtamedullary nephrons. Okay. So here's their difference. This one is called cortical nephron because most of that nephron stays inside the cortical region. Juxtamedullary nephron has a long loop of handling that dips down into the renal medulla. Okay, you see here, got a long loop of handling. Okay. So even though the cortical nephron also has a loop of handling that somewhat dips into the renal medulla, but it dips in only slightly compared to this long loop of handling. And this minority of nephrons is important, particularly important, and contributing to concentrating urine, forming a really concentrated urine. When you form a concentrated urine, you're helping your body retain water. Okay, a urine is concentrated because most of the water is being pulled out. Okay. Now I want to do a quick review of what a portal system is. So during the blood vessel chapter, we talked about what a portal system is. Right? And then when we got to digestive system, we talked about the hepatic portal system. So recall that the hepatic portal system, or portal system in general, it's a vascular system that consists of two capillary beds. Most of the time, each of our organ or our tissue gets supplied with a single capillary bed that then drains into the venous side of your blood vessels. Okay, so it takes the arterial blood that's oxygenated, and then in that capillary bed that supplies the tissue, those oxygen gets used up, and then that capillary bed drains into the venous system. However, in the portal system, there are two capillary beds. So this is what defines the portal system. There are two capillary beds. Specifically in the hepatic portal system, the first capillary bed is located inside your intestines. That's the capillary bed that allows for nutrient absorption. Okay, nutrient absorption as well as toxin absorption. The alcohol that you drink and stuff, that has to first pass through the second capillary bed inside the liver to be detoxified. Okay, and some of the nutrients is put away and stored there before that blood can re-enter into the general circulation, into the venous blood. Okay. In the renal, in our kidneys, we also have a similar schematic. There are two capillary beds involved in the renal portal system. The first of which is the glomerulus. Notice that when the blood passes through the glomerulus, it's not deoxygenated yet. It's simply undergoing filtration. Then this blood continues into the peritubular capillaries. This is the second capillary bed. So this is the renal portal system. And the renal portal system, okay, can also involve the vasa recta. Vasa recta are these specialized blood vessels that are intimately wrapped around the long loop of handling of these juxtamedullary nephrons. Okay. Students have seen pictures that look slightly different from this. I want to mention that the peritubular capillaries can either lead directly back to the cortical radiate veins, or it can lead into these vasa recta before draining into the cortical radiate veins. What is faulty about this picture is that it should have shown peritubular capillaries here as well. Okay. So make a note of that in your lecture note, is that whether it's cortical nephron or juxtamedullary nephron, both are going to have peritubular capillaries. But in the case of juxtamedullary nephron, those peritubular capillaries will then lead into vasa recta, which then drains into the cortical radiate vein. Okay, I should have also mentioned earlier that cortical radiate arteries and cortical radiate vein has a different term called the interlobular veins and arteries. Okay, interlobular arteries and veins. Okay, let me just put that up here and I'll get back to you in just a second. Please do not confuse interlobular with interlobar. Okay, these are considered little cortical lobules. Okay, whereas this is a lobe. So interlobar is between the lobes. Interlobular is between the lobules. These peritubular capillaries, they pick up the things that have been reabsorbed through the renal tubules. And then through the peritubular capillaries, these substances can then be returned eventually back into the general circulation. It's also through these peritubular capillaries, things like some of the toxins that or medications that or drugs that you ingest, they are renally excreted because they can either be filtered through the glomerulus, or if they're not filtered through the glomerulus, they may be secreted. Okay, meaning they're first dumped by the peritubular capillaries and then picked up through the tubular walls and then dumped into the tubules. Okay. And then eventually the tubules will then drain into the collecting duct and then out of your body eventually, okay? So go over this part on your own. Okay, so, okay, that's what we're gonna talk about. I'm gonna take a little break. The juxtaglomerular complex, also known as juxtaglomerular apparatus, let me say that again, juxtaglomerular apparatus, JGA or JGC. This is a structure that involves your glomerulus or your renal corpuscle making a contact with the distal convoluted tubule. And this structure is important in the regulation of pressure, blood pressure. And that's something you're going to learn about in physiology. But we want to look at the structure of this JGA. Okay. So if you look at each of these nephron pictures closely, you'll see that. So this is the proximal convoluted tubule. Okay, and then after the loop of Henle, it leads into the distal convoluted tubule. Okay, and this distal convoluted tubule eventually makes it near this renal corpuscle and makes contact with it. And it's a stem here, okay? So if we take a closer look at that, the cross-section of that will look like this. Okay, so this is a section, the cross-section of that distal convoluted tubule. And this is a cross-section of the afferent arteriole, efferent arteriole, okay, and then the renal corpuscle. You can see that this DCT, okay, is affiliated, winds back and associates with the afferent arteriole and efferent arteriole. Okay, so this is known, this association, this is known as a juxtaglomerular complex or apparatus. In this structure, these cells located on the, some textbook will say the end of the ascending limb loop of Henle. Some textbook will say it's the beginning part of the distal convoluted tubule. Okay, either one is fine on a test. I'm not gonna have you pick between the two as multiple choice, right? Now, macula densa cells are these cells that are located in this structure and lining on the side of the ascending limb loop of Henle. Okay. And then there's extraglomerular mesangial cells, or just called mesangial cells. Okay, that are in between the macula densa cells and the granular cells. The granular cells are cells that are along the wall of this afferent arteriole. Okay. And these granular cells are important in secretion of a hormone called renin, which also plays a role in blood pressure control. So this whole structure is very important in blood pressure control. I won't go into the detail because that's something you're going to go into extensive detail about when you get to physiology. Okay. I want to make a brief mention that these granular cells, they are specialized smooth muscle cells of the afferent arteriole. So you have other parts of afferent arteriole all lined with smooth muscle cells, but once you get here, you can see the appearance of it in the illustration. It looks different, right? So this part of the afferent arteriole has a modified type of smooth muscle cells. And those cells specifically are called granular cells. And the granular cells have another term for them. They're also called the juxtaglomerular cells, or JG cells. Yeah, I'll make sure to make a note of that in the notes. So if you got through what we just talked about in the lecture, this part will seem a lot easier. After the kidneys, the urine is then going to drain down the collecting duct, correct? Okay. And so the collecting ducts, many collecting ducts will then merge and they will drain into renal papilla. Okay. And the renal papilla is a tip of these renal pyramids. So you can imagine these lines here we see, these are collecting ducts. Collecting ducts really span from cortex into the medulla. Okay, so when you look at this image, I want you to understand what is in the renal cortex, what is in the renal medulla. Okay. The cortex has the glomerulus, the renal corpuscle, it's got PCT, DCT, but then most of the loop of Henle is going to be in the medulla. Okay, whereas the collecting duct goes all the way from the cortex into the medulla. And these collecting ducts are going to merge into the tip. This tip is called the papilla of the pyramid, or pyramid papilla, or you could, as long as you got those two terms in there, I'm good. Okay, so papilla of the pyramid is the tip. The papilla then drains that urine into minor calyx, or minor calyces, okay, for plural. Minor calyces lead into major calyces. So this is the major calyx. Major calyces then drain into the renal pelvis. Okay, so calyx and the renal pelvis. Okay, the renal pelvis then dumps its urine into the ureter, and that's where we are. The ureters are actually muscular structures. It's got smooth muscles in there. So when you get a kidney stone that gets stuck in here, your ureters can actually spasm trying to squeeze that stones through. It's got two layers of muscularis, longitudinal layer and circular layer. Okay, so notice the layers. Remember from the first unit that ureter, as well as the inner lining of ureter as well as bladder, okay, is lined with transitional epithelium. Okay, this type of epithelium allows for stretching. So unlike in the digestive tract, that smooth muscle layers has its inner layer being longitudinal, whereas the external layer is the circular one. You don't have to worry about this. I'm going to cross this out. Okay. Now the urinary bladder is a very muscular structure. It's got a thick layer of smooth muscles in it. And that muscle is called the detrusor muscle. Detrusor. Okay. Detrusor muscle. So it's a very distensible muscular sac. The inner lining is transitional epithelium that's surrounded by connective tissue, as well as a smooth muscle, a thick smooth muscle layer called the detrusor muscle. I'm going to have you cross this out. Okay. Most of the nervous fibers, neurons that are supplying to your detrusor muscle is parasympathetic fibers. And that allows for contraction of your bladder. So when your bladder is filled up, it starts contracting. That causes that sense of urgency. Okay. So in a full adult urinary bladder can hold approximately 500 milliliters of urine. Now, if we look at the trigon is this elevated triangle on the inner surface of the posterior wall of your bladder, and it's defined by the ureteric and urethral openings. So here are your ureteric orifices. This is where those ureters, the urine inside the ureters exit the ureter and enter into the bladder. So these are ureteric orifice. Okay, this is where this internal urethral sphincter is. Okay, that forms a triangular region, raised triangular region with the ureteric orifices. So this is called the trigon. Finally, the urethra. The urethra, remember, it's to be differentiated from ureters. Urethra is quite a different structure between men and between what they're like in women. But in general, it's going to be this tube that drains your urine out of your urinary bladder and then transports it out of the body. In men have a much longer urethra, and actually their urethra is so long it's divided into several segments. The part that passes through the prostate, so this is the prostate gland, the part of urethra that passes through the prostate gland is known as prostatic urethra. And then the part that passes through this urogenital diaphragm is called the intermediate urethra. And there's a prostatic urethra, intermediate urethra, and then this is the spongy urethra that passes through the penis. And then the urine then exits the external urethral orifice into wherever, hopefully it's a toilet. Okay. You can see that in females have a much shorter urethra, and this is the primary cause of a much higher frequency of urinary tract infections in women. So that E. coli or whatever bacteria only has to ascend, okay, it only has to travel such a short distance before they enter into the bladder. Okay. So this is the reason why women tend to, it's not uncommon for, most women have experienced at least one UTI in their lifetime. But it's a lot less common to see men with UTI. Understand where the external and internal urethral sphincters are located. Okay. And also understand that the type of histology in here, this is transitional epithelium. Okay. Eventually, near the end of this urethra, the transitional epithelium becomes stratified squamous. I'm blanking out. So it's going to be the non-keratinized stratified squamous epithelium. Okay, because this is a wet surface, a wet membrane, right? So it would not be keratinized. So non-keratinized stratified squamous epithelium. We can forget about this part.